Hi, welcome back to My Smart Learning. It's me, Mr. Meehan, with your A-level biology content. We're finishing off photosynthesis today. This is for year 12s and year 13s and year 11s if they're interested in doing A-level biology, if you're doing separate biology at the moment. Limiting fact, or you could be doing your trilogy as well, you can still do it. Right, so today we're looking at limiting factors to finish it all off. It's kind of basic GCSE stuff, to be honest. Question then, can you make plants grow faster? Well, obviously you can, because that's what farmers do. They try to maximize their yield out of their plants. They want them to grow faster, they want them to grow bigger, which means they need to do lots of more photosynthesis. Right, so recall quiz then. Obviously you want your high five because you can't buy those in the shops, not that you can go to the shops anymore. What are the three stages of photosynthesis? Number 2. What are the products of the light-dependent reaction? Number 3, what are the products of the light-independent reaction? Number 4, what are the tissue layers of a leaf? And the last question, what are the limiting factors for photosynthesis? What do you know already? So have a go at those questions, pause the video, give them a go, and when you're ready for the answers, you can unpause, like now. Right, so number 1, what are the 3 stages of photosynthesis? You've got the light capturing stage, you've got the light, I'm giving the answers here, the light dependent reaction, and the last stage is the light independent reaction, also known as the Calvin cycle. What are the products of the light dependent reaction? Well, the products of the light dependent reaction, you get oxygen as a byproduct, but you also need these 2 things. You get NADPH, also known as reduced NADP, and you get ATP. And what are the products of the light independent reaction? So from the light independent reaction, well, we mean products, or what are the things inside of it. You get RUBP, ribulose bisphosphate, carbon dioxide joins onto it. You get GP, which is 3GP, glycerate phosphate. You get from that your triose phosphates, and then you end up with those triose phosphates eventually turning into things like organic compounds like glucose. So what are the limiting factors? No, sorry, missed one out. What are the tissue layers of the leaf? We talked about this last couple of lessons back. We got your cuticle, upper layer and bottom layer, but then you've got your upper and lower epidermis. Then you've got your mesophyll layers, which is your palisade mesophyll and your spongy mesophyll. In your lower epidermis, you've got your stomata, which are formed because of the guard cells, and also you've got your vascular bundle, which has got your phloem and your xylem tissues. And then what are the limiting factors for photosynthesis? So this is GCSE level. What do you know? Well, what can speed up photosynthesis? Well, carbon dioxide, light intensity, temperature are your 3 main ones you need to know. But then you've also got the biological aspects as well. Those are physical abiotic factors. The biotic factor, or the biological factor here, would be the amount of chlorophyll, or the number of chloroplasts that you have. So the more chlorophyll, the more photosynthesis. So that could be a limiting factor as well. So we're going to be focusing on those first 3 abiotic factors. How do light, temperature and carbon dioxide affect the rate of photosynthesis? How can I measure the rate of photosynthesis? Is what you need to know. So it is actually the required practical in GCSE and A-level biology. This is like a basic model of it. You got a beaker, you put some pondweed in there like Elodea. You can have a inverted upside down glass funnel or transparent funnel, and you submerge it completely in water. You have the whole thing with water, and as the photosynthesis is happening in the Elodea, bubbles of oxygen is given off, and you can capture the oxygen. You can either make little lines in there, so like some kind of gradation on it, or you could have a measuring cylinder, and you can measure the volume of oxygen being produced over a given time. You can shine a light on it, and you can change the light intensity either by turning up the voltage on the bulb, making it brighter, or you can move the light bulb closer or further away and have a ruler, a meter ruler, measuring the distance the light is away from the plant. You could change the amount of carbon dioxide though, if you want to change, because the light is a limiting factor. The amount of carbon dioxide can be changed by adding hydrogen carbonate. Now hydrogen carbonate is just baking soda, but when you dissolve it in water, it's highly soluble, it just dissociates and breaks down and makes carbon dioxide. So the number of spatulas or the grams of hydrogen carbonate will affect the amount of carbon dioxide in that solution. And also the third one, you can change the temperature by just taking the temperature of the water by heating some water in a kettle and then using a thermometer, taking the temperature of the water that way, or having it set up in a water bath. Okay, that's like a basic child's play little system. How can I make it more technical? How can I make it so I get more accurate and more valid data? So, how can you measure the rate of photosynthesis? It's like measuring any rate, something over time. And you either measure how quickly the reactants are being used up or how quickly the products are being made. From here, it's how quickly the products are being made. So you can either work out how quickly the carbon dioxide is being used up, but that's difficult. How do you measure that? How do you see how much is actually being used and taken up? It's too hard. So what's easier, the second way, is the release of oxygen. So the bubbles of oxygen given off. That is a much simpler method of measuring the products being produced and therefore the rate of photosynthesis. So we've got a photosynthometer. Now photosynthometer is kind of the opposite of a potometer. And remember, a potometer sucks the water up or draws the water up and the bubble moves along with it, and you can measure the rate of water uptake in a plant. This is kind of the opposite way around. So this is a photosynthometer, as in a photosynthesis meter. Okay, so I got my pond weed, it's got my shoot of Eladia. Keep it in the pond water with some hydrogen carbonate, so that gives it the amount of carbon dioxide. Now that could be my limiting factor, so I can change the amount of that if I wanted to. Put it in a water bath so that it stays warm, so the plant doesn't go into shock, and then gives it an ambient temperature. Got a thermometer to control that variable, so it's a valid comparison. Plants can produce oxygen, obviously. So what will happen, the streams of oxygen will go up, there's a capillary tube, that bubble, the gas can be trapped and captured and drawn through this, and we've got a scale here in millimeters, so we can measure the volume of oxygen be given off in a given time. Well, how would I measure the actual volume? Because I can just get a stopwatch and measure it over 5 minutes, 10 minutes, an hour, whatever, and measure how far the bubble has moved. That's a quick, simple way, and that's a comparison from one variable to another. So I can change the amount of CO2, change the temperature, I can change the amount of light intensity and see how far the bubble goes compared to the other times. So that's a valid comparison. However, how would I measure the actual volume of oxygen? Well, I need to capture every single bubble that comes through there. So therefore, if I'm drawing that up and I measure the actual volume, that's a capillary tube. The capillary tube, the shape of it is a cylinder. Now, how do you work out the volume of a cylinder? The volume of a cylinder is how do you work out the volume of a cylinder? It's pi r squared, which is the face, surface area, times the distance it's moved, or the length. So pi r squared times the length. So therefore, you can work out the actual volume of oxygen produced over a given time, and that'll give you more accurate data to work on. So, and that's it, and you can push it back with that little, there's a capillary tube, you've got a syringe there, so if you are just measuring this bubble, pushing it, and then measuring how far it goes, you can just push it back and set it back to 0 to restart this experiment. So that's a photosynthometer. What kinds of data can you get? Well, you usually get, for a rate of reaction or rate of photosynthesis, you get a line going up and then it starts to plateau because it hits what we get limiting factors. So you keep going faster, 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 and there's something that's becoming a limiting factor because there's not enough of something else. So the kind of obvious shape is that you get a straight line going up, because the more light I give it, the faster, the faster, the faster, the faster, the faster the rate of photosynthesis, and then it starts to plateau, because no matter how much light I give it, it's not going to get any faster because something else is limiting it. So it could be there's not enough carbon dioxide. So therefore, if this was in a low carbon dioxide environment, it can't go any faster because there's just not enough reactant to use. It's using up the reactant so quick, there's none left. So therefore, if I give it more CO2 on top, then the graph will look the same like this, but it will go up further and then plateau further along the top, because this is what we know as limiting factors. Something else is limiting. It could be the lack of chlorophyll molecules. There's always going to be something that there isn't enough of. So I always use this analogy of Formula 1 cars. So if you think about Formula 1 cars, they go really, really, really fast, but they hit top speed because there's always something that's not enough of that you can tweak to make it that little bit faster. So for instance, you can reduce the mass or the weight of the car. You can make it more aerodynamic. You can make it have a bigger engine. You can change the tires. You can, all sorts of things you can do, and that's what the whole point of Formula 1 is they're trying to maximize efficiency and speed of a car and handling of a car and race it so that they want to get it faster and faster. And it's all about tweaking, because there's always something that there's not enough of. There might be the maximum of something, but something else is holding you back. Okay, it's usually with cars. It's usually drag. Okay, you can't get any faster because you've got air resistance going in the opposite direction. So you need to streamline the shape. And then obviously it's the weight and so on and so on. So same thing for photosynthesis. There'll always be something else. So if I've got loads of light, there might not be enough CO2, or the temperature, it might not be the right temperature, or there might not be enough chlorophyll molecules. So there'll always be something, there'll always be one thing that's holding it back from going any faster. Okay, and we call that the limiting factor. So the compensation point, what do I mean by that? The compensation point is, I use the analogy of like break-even analysis when it comes to running businesses and things. So if you imagine you're a business, you're going to spend money to start your business up. And so that means you're using money before any profit's being made. So until you hit that sweet spot, or that bit where the amount of money you're putting in and the amount of money coming out, when it's even, we call that break-even analysis. That means the net will be zero. So the amount of money coming in and the amount of going out is zero. So there's no money being made. After that is when you become profitable. So you start to make more money than you've put into the business. So therefore, you've broken even. Broken even is when you get zero. So then you start to become making profit, gross profit. So the compensation point's a bit like that. At night time, are plants photosynthesizing? Well, no, because there's no light. So plants use carbon dioxide, obviously, for photosynthesis. However, they also give out carbon dioxide. If you didn't know that, then it's a bit shock, spoiler alert, plants make carbon dioxide. How? They make carbon dioxide because they are respiring. Just like you and me, they're respiring. They're aerobic organisms. Like, there, I give out carbon dioxide. Now, you don't see plants breathing out, but plants do give out carbon dioxide. At nighttime more, because they're not photosynthesizing, so any respiration will be giving out carbon dioxide. So they're giving out carbon dioxide and using up the oxygen. Any surplus that they've made or any in the atmosphere, they'll be using the oxygen at nighttime. However, the amazing thing is the sun has got its hat on, like today, hip 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 hooray. The sun will start to rise, and as the sun rises, this is the morning, the sun will start to rise. As the sun rises, okay, so from here the sun starts to come up. As the sun rises in dawn, the amount of carbon dioxide given out starts to reduce because the plant starts to photosynthesize. And as the plant starts to photosynthesize, it's starting to produce carbon dioxide rather than taking it in. When it hits that sweet spot, that zero, we call that the compensation point. At that point, the amount of CO2 that it's giving out from respiration and the amount of CO2 it's taking in from the atmosphere for photosynthesis has hit a net zero. It's balanced out. It's at zero there. So the amount going in and coming out is exactly zero. So therefore, when the sun starts to shine, there's more sunlight, more and more carbon dioxide is actually taken in, it's more of it is taken up because it's absorbing it for photosynthesis, and less is coming out because it's still respiring. Plants are still respiring. They don't suddenly switch off the respiration because, oh look, I've got photosynthesis, I can stop breathing, I'll stop respiring. Obviously they're respiring. You respire 24/7. You only photosynthesize during daylight. But the net effect is you're producing a lot more oxygen than you're actually using. So it's surplus, so you're giving out more oxygen. Carbon dioxide, you use up more carbon dioxide than you're actually producing. So you're absorbing, you're a net consumer. You're a net consumer of carbon dioxide. A plant is obviously not me. Right, so, and that's what that means by the compensation point. And that happens at dawn. But then eventually, more light, photosynthesis gets faster until it starts to plateau. So what's the biology behind it then? So light intensity is increased, more carbon dioxide is absorbed, more oxygen is produced until they are both balanced. This is known as light compensation point, like we said. If the light is increased further, it becomes proportionally increases the volume of oxygen given out and the carbon dioxide taken up. Finally, it plateaus where it can't go up anymore because we've hit a limiting factor. So here you've got the low CO2, so that it plateaus sooner. Here you've got a higher CO2, it plateaus further up. but the temperature is still low. Make it warmer and higher CO2, we get a much higher rate of photosynthesis, but then it plateaus still towards the end. There's still something, a limiting factor. This time it could be the amount of chlorophyll. Not enough chlorophyll, not enough chloroplasts, plants too small, it can't literally, take in as much light as possible. So, data then, what does it look like for carbon dioxide? So the more carbon dioxide, the higher the concentration of carbon dioxide, you can see the colours, the higher the concentration, the faster the rate of photosynthesis. And why does it look like that for temperature? Well, for carbon dioxide, obviously it's a reactant, so the more reactant you give it, the more reactant you got, the more product you can make it faster. So you get a similar graph to this. So if this wasn't carbon dioxide, if this wasn't light intensity, but it was carbon dioxide, so you had CO2 increases here, you still get the same shape graph. What it will say is, it'll say low light, medium light, high light. Okay, so you get the same shape. However, with temperature though, you get this bell-shaped curve. So it goes up, then it hits a peak, and it shoots right down straight after. So why is that? Well, it hits the optimum. So the biology behind that then is the rate of photosynthesis increases, the higher the temperature, the more kinetic energy the particles have. The kinetic energy for the reactants, for instance carbon dioxide and the water, the more collisions you're going to get, the enzymes, they're going to move around more freely, so you're going to get the activation energy will be easier to reach. Therefore, reactions going to happen faster, photosynthesis is going to be much faster. So the kinetic theory, it hits the optimum, the peak, the hottest part. But what happens to enzymes when they get too hot? That's right, they don't die. If you say that, then somebody needs to die. But enzymes denature, that's the word we use. Enzymes don't die, they are proteins, they're not living things. Enzymes denature, so that's what happens beyond the optimum. Enzymes denature, which means the active site changes shape. The chemical, the heat energy, the thermal energy will break those bonds, distort the structure of active sites and the structure of the proteins. Therefore, enzyme substrate complexes can't be made. Therefore, the reactions can't continue. The rate of photosynthesis goes down. Eventually, the plant will die. So I've got some links for you guys to catch up with all your photosynthesis stuff. There is a ton of stuff on Caboodle. There's animations, PowerPoints, all sorts of stuff, and also obviously on YouTube. I'm not the only person on YouTube, obviously. But hopefully you've learned something. This is the end of the photosynthesis topic. So this week we are done with all of chapter 11. So it's amazing. We've started the year 13 work. We're probably going through work faster than we would have done when we were at school. So lockdown may have some benefits somehow, weirdly. But hopefully you've learned something. If you have, then please watch them all, like them all, share them all, subscribe if you haven't done so already. And I forgot to give you your virtual high five. You can't buy those in the shop. So without further ado, your virtual high five. See you next time.